Welcome to this video where we're going to be looking at our PowerPoint here covering the different factors that can affect cellular respiration. So specifically out of the study design, the key knowledge points we'll be looking at are these two. The first is the general factors that impact on enzyme function in relation to photosynthesis and cellular respiration. We're only going to look at it in this one in relationship to cellular respiration. And those are the changes in temperature, pH, concentration, competitive and non-competitive enzyme inhibitors. And then also the factors that affect the rate of cellular respiration, temperature, glucose availability and oxygen concentration. So those are the things we're going to be looking at here. So just a quick recap around cellular respiration. When we're talking about aerobic cellular respiration, there are three key stages: glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis is in the cytosol, the Krebs cycle, and the electron transport chains both occur in the mitochondria. Overall, you can see our reaction is glucose plus oxygen gives you carbon dioxide and water. So our inputs are glucose and oxygen, and water and carbon dioxide are our outputs. And so you can also see our diagram there just showing where those different stages occur. Now, in terms of factors affecting cellular respiration, there are plenty of different factors that affect how efficiently cellular respiration is able to work. And we've grouped them here into substrate concentration, either glucose or oxygen availability, temperature, pH, and the different types of competitive and non-competitive inhibitors. Now, how exactly each of these factors can impact on cellular respiration depends on whether or not you're talking about increasing the factor or decreasing the factor. And we're going to go through and look at each of them in turn. So first we're going to look at substrate concentration. And this graph might look fairly familiar, and there's going to be a number of things in here that will look familiar because they're very similar effects to what happens when we make these changes with photosynthesis. But we are going to specifically look at here cellular respiration. So like with any chemical reaction, when you have changes in the level of reactants to substrates, that can have an impact on the reaction itself. Now, cellular respiration, all the reactions that are involved in there get catalyzed by enzymes. So instead of, like we might say traditionally with chemistry, talking about reactants for a reaction, we more correctly would use the word substrate. So be aware that here we're sort of using reactant and substrate interchangeably, but if you're talking about something that's picked up by an enzyme, substrate is more correct. Now, cellular respiration has two inputs, therefore two reactants or two substrates, and those are glucose and oxygen. So if we alter the levels of glucose or oxygen, we can have an impact on the rate of cellular respiration, how quickly it is happening. So first of all, if we're looking at glucose availability, how much glucose is there? Now, generally speaking, as glucose concentration increases, the rate of cellular respiration increases until all the enzymes that are being used actually reach what we call a saturation point. And that's just where all of those enzymes are in use, and you can keep adding glucose, but it won't make any difference because you're going to still have to wait till those enzymes become available because otherwise they're saturated. Now, glucose is actually the input at glycolysis, and so we're looking at the enzymes involved in glycolysis becoming saturated. Now, that is the first stage for both aerobic respiration and what we call anaerobic fermentation. So both of these reactions, which sort of finalize afterwards after glycolysis, both of those require glucose to go through glycolysis at the start. What then happens afterwards is dependent on oxygen availability, but both of these are relying on glucose being available. So if you don't have glucose available, neither aerobic or anaerobic respiration could occur. Something that cells will do to help make sure that they always have the glucose they need is they actually, in times where they've got excess glucose, will store that excess glucose. Animals will store it initially as glycogen. They then have other mechanisms for storing things, and plants will store it as starch. And starch is actually made up of lots and lots of glucose molecules joined together into forming a polysaccharide, so a very long chain of glucose. And what that means is those cells, when they're experiencing times of low glucose levels, they can actually go back to these molecules, this glycogen and this starch, and they can break them down to free up that glucose and release it back into the system so that their cells now have increased their glucose levels to the point that they need to function properly. So that is the first thing with how glucose availability can affect cellular respiration. Obviously, low glucose, low rate of reaction, high glucose, higher rate of reaction, and it will increase as you increase it to the point where it hits its maximum rate. This brings us to exam question 1. So exam question 1 says, the apparatus shown on the right was used in a series of experiments to study aerobic respiration. In 3 different experiments, the reaction tube initially contained the following: 1. Suspension of mitochondria, 2. Cytosol of cells from which the mitochondria had been removed, or 3. Suspension of mitochondria and cytosol of cells. The temperature and pHs of the mixtures within the reaction tubes were carefully controlled so as to not damage the mitochondrial structure or any of the enzymes. In each experiment, a solution containing glucose was first added to the mixture in the reaction tube, and the oxygen concentration was measured for 3 minutes. Then a pyruvate solution was added, and the oxygen concentration was measured again for 3 minutes. Using your knowledge and understanding of aerobic respiration and mitochondria, complete the tables in the next slide with your prediction of the change in oxygen concentration of the mixture in the reaction tube after the addition of each substance, and give your reason for your prediction. So what you need to do in a moment is you'll need to fill in these tables, and you can see you've got your 3 different experiments, and those relate to these 3 different tubes that you've got listed there and what's in them. And for each one, you need to say, what do you think will happen to the oxygen concentration when you add glucose? Do you think it will increase, decrease, or have no change? And what do you think will happen when pyruvate is added? Okay, increase, decrease, or no change. So you need to go through and make your predictions, and for each prediction you make, you need to justify it and give a reason for why you think that is the case. So you can pause the video now as you go and answer the question, and once you've answered it, go and watch the video linked in the description box below to check out how to answer this question and correct your work. So we've looked now at how glucose availability affects aerobic respiration. We're going to also look at oxygen and how it affects aerobic respiration. And we'll also look at it in relationship to anaerobic respiration. So obviously, if you don't have oxygen, you're going to have some problems with aerobic cellular respiration, because aerobic cellular respiration requires oxygen as an input. So aerobic respiration requires oxygen for the electron transport chain to function. One of the key points is the electron transport chain is going to take the oxygen from those oxygen molecules, and it is going to combine it with hydrogen to form water as its end product, or as one of its end products. So if you don't have oxygen, that's not going to work. Now, as oxygen levels rise, the rate of aerobic respiration can increase with it. Now, when oxygen is low, some cells will actually switch to anaerobic respiration. And it's normally not a prolonged thing. They'll do it until oxygen becomes available sufficiently again. So generally speaking, low oxygen means a low rate of aerobic respiration, and possibly even no aerobic respiration, only anaerobic. Whereas high oxygen generally means high rate of aerobic respiration. And that's what we tend to expect when we change oxygen levels and we look at the rate of aerobic respiration. Now, there is something to consider here, and that's at a certain point. If you have unlimited glucose, you can keep adding oxygen, but it's not actually going to increase beyond a certain point, and that's just got to do with the enzymes involved being saturated again. So you need to keep in the back of your mind that because you have enzymes that are catalyzing these reactions, once they get saturated, you've hit your maximum rate of reaction. Okay, and the only way that you could increase that is by adding more of those enzymes, and that's not something we tend to consider. We tend to look just more at the substrates, so the oxygen and the glucose in question here. Now, with anaerobic respiration, it's worth noting that if you have low oxygen, there are certain organisms that will do anaerobic respiration instead. But there are also certain organisms that only respire anaerobically. Now, this is just sort of something to keep in the back of your head, but it's not a super common thing. And that's just that you have some organisms that they actually don't want to respire aerobically. And so if you're talking about the presence or absence of oxygen, they're going to be affected a little bit differently to humans or other animals and that sort of thing, or anything that actually wants to do aerobic respiration. So organisms that only respire anaerobically get affected in 2 ways when oxygen is present. Either not really any response, it'll be very minimal, and they're just going to keep doing anaerobic respiration as they normally do. So they just ignore that oxygen, basically. Or there are actually some organisms that would die if the oxygen is present. So these organisms either have no reaction or a negative response to the presence of oxygen, and so oxygen availability actually affects them a little bit differently to organisms that can respire aerobically. So for this small group of organisms, no oxygen means a high rate of anaerobic respiration, whereas oxygen present means the rate of anaerobic respiration will either be the same, lower, or none, because you've killed the organism. Now, there is a diagram there. Just really want to emphasize, you do not need to know those terms. That's just there as a example that we have a range of different organisms where some need oxygen, obligate anaerobes, some obligate anaerobes, and those for them oxygen is actually toxic. You have some that is facultative anaerobes, and those prefer oxygen. They'll go without if they have to, but they prefer having it. And there are aerotolerant anaerobes, and those ones put up with oxygen present. They'll keep doing anaerobic respiration, but they will deal with the oxygen if it's there, and they'll just sort of ignore it. So this is a much smaller group. Generally speaking, we're more focused on what happens to aerobic respiration when oxygen is present, but there is a small group of organisms where they're normally anaerobic, and so oxygen presence has a slightly different effect on them. And it's worth keeping that in the back of your head, but it's not a major point to consider here. This brings us to exam question 2. Exam question 2 says the rate of aerobic cellular respiration in a human cell may increase if the A. Temperature of the cell is lower from 37 degrees Celsius to 35 degrees Celsius. B. Oxygen concentration available to the mitochondria increases. C. Carbon dioxide concentration in the cytosol of the cell increases. Or D. Rate of facilitated diffusion of glucose into the cytosol of the cell decreases. So you can pause the video now as you go and answer that question, and once you're done, go check your answer against the answer video linked in the description box below. So this now brings us to temperature and how temperature affects cellular respiration. So at lower temperatures, pretty much with any chemical reaction, your rate of reaction is slower. And that's just because at low temperatures, the molecules involved do not have as much energy, and that's kinetic energy. So if they don't have much kinetic energy, they're not moving around very much. If they don't move around very much, then they're less likely to collide and react with each other. So that's why low temperatures end up giving you a lower rate of reaction. Some animals actually, because it's that those rates of reaction are lower, will actually want to sort of hibernate and avoid needing as much energy, needing this reaction to occur as much. So they reduce their energy requirements, which means if the reactions are a bit slower, it doesn't affect them as badly as if their energy requirements were as normal. Now, if you increase the temperature, you increase the amount of energy those molecules have. They move around more. The enzymes and substrates are more likely to collide. Therefore, the reactions are more likely to happen. So the rate of reaction increases. And in this case, we mean the rate of respiration increases. Now, remember, cellular respiration is being catalyzed by enzymes, which are a type of protein. And we know that with proteins, if we increase the temperature, they will have a certain temperature at which they denature. Above that temperature, they will become denatured. And if they denature, they will start to lose function rapidly to the point where they'll be non-functional. So it is worth noting that we kind of get this slow increasing sort of graph to its optimum temperature where it peaks at the top, and then a quick drop off as you continue above that optimum temperature where the protein is becoming denatured. And so that is what we can see in the diagram there. So as with temperature, its impact on the rate of respiration is that if you have low temperatures, it is a lower rate of reaction, and as you increase it, the rate of reaction increases to the optimum temperature where it's going as fast as it can, and then beyond that, if you keep increasing it, you'll cause the enzymes to denature and the rate of reaction will rapidly drop off. So we end up with that sort of graph there. Now with pH looks a lot like temperature, but kind of, there's no slow increase. It's a quick increase and a quick drop off as you go from a low pH to a high pH. Now, the exact peak of that optimal pH is going to vary dependent on the enzyme and where it's normally located. But like with photosynthesis or anything where we have enzymes catalyzing these reactions, you have proteins, which are the enzymes doing the reactions, and pH can also cause them to denature. A pH that is too low or a pH that is too high will cause them to denature. So the optimal range is somewhere specific to them. And so we get this sort of bump looking graph where we have no function sort of at the very low end, no function at the very high end, and then we have sort of this quick increase to the optimal and quick decrease once we go past the optimal. And so it's a very symmetrical looking graph compared to the graph of temperature, which has got a long slope on one side and a quick drop off on the other. But here we see the same thing. There is an optimal pH where the enzyme is going to function best, either too high or too low around that, and you're going to denature that enzyme, and therefore it will not function well, if at all. And so that is how cellular respiration is going to get affected by the pH. So Like I said, different enzymes will have different optimum pHs, and you can see here in the graph that you have enzyme A and enzyme B, and they've got different optimum pHs, and that probably means they exist in different environments, where A is in a slightly more acidic environment and B is in a slightly more basic environment, and that's what they enjoy and where they function best. And so for the enzymes involved in cellular respiration, the cytoplasm of a cell tends to have a pH around 7.2. So the enzymes that do glycolysis are going to function best around that pH. Whereas in the mitochondria, if you look at the intermembrane space, it's from 7 to 7.4, whereas the matrix is actually a bit higher as 7.8. So anything involved in reactions in the Krebs cycle, where it's happening in the matrix, should be able to work well around 7.8, which is higher than what it is in the cytoplasm. So you might actually see that these enzymes that are involved in these different stages of cellular respiration actually end up with slightly different optimum pHs, just based on where they're meant to be found and where they're meant to be functioning. So this brings us to inhibitors, and we've addressed inhibitors before. We know that there are two types of inhibitors: competitive inhibitors, where the enzyme attaches to the active site and will actively block the substrate from attaching, versus non-competitive inhibitors, where they'll attach somewhere else to the enzyme, cause a change to the shape of the active site, and that stops the substrate from binding. So in general, if you've got inhibitors are present and they're attached to the enzymes involved in cellular respiration, they're going to lower the rate of cellular respiration, simply because they're stopping the substrate from binding and being catalyzed by that enzyme, and therefore the reaction doesn't go ahead. So we can expect with inhibitors that they will generally decrease the rate of cellular respiration. Now, some inhibitors are bad. In fact, generally when we think about inhibitors, they're bad, but there are times in cellular respiration where there might actually be a form of good inhibition. So any inhibitor that affects the metabolic pathways of either aerobic respiration or anaerobic respiration have the potential to be incredibly bad. And that's generally, like I said, what we think about. And so that could be how cyanide is a non-competitive inhibitor of cytochrome C in the electron transport chain, and it will completely disrupt that, which is a big problem. And it will stop the electrons being transferred along the electron transport chain pathway, and it will stop the protons from being able to move back from the intermembrane space, and that's going to stop ATP synthase from being able to make ATP. So that's really, really bad, and that's going to be very detrimental to the cell and the organism as a whole. But there are times when the inhibitor can actually be really beneficial. And ATP, for instance, can actually act as a non-competitive inhibitor of an enzyme called 𝗂𝗂𝗺 and that's an enzyme involved in glycolysis. And what it means is ATP will bind to this enzyme and actually stop it from functioning in glycolysis. And the reason we want that is you only want glycolysis happening if you're actually going to need ATP to be produced. So if you have lots of ATP, you don't want glycolysis to get started and then to produce stuff for the aerobic respiration pathway. So if we can pause glycolysis when we've got lots of ATP, that's really great. It means we're being efficient and not wasteful of things, and we're not doing reactions when we don't need them to happen. So when ATP levels are actually high and you don't want glycolysis occurring, that ATP will actually end up binding to the 𝗂𝗂𝗺 and it will actually stop it from doing glycolysis. That's really good. This is something you call end product inhibition, and it just means that the end product of this pathway, when you've made a lot of it, will actually end up inhibiting some of the initial enzymes, just to basically put a pause on that reaction from happening. Now obviously as that ATP gets used up in the cell, it's going to end up detaching from that enzyme, which will allow the enzyme to refunction again in glycolysis, and then it'll produce the pyruvate through glycolysis, the acetyl coenzyme A, it'll go through the Krebs cycle, the electron transport chain, and it'll start producing lots of ATP again, and the ATP levels will increase, and they may end up rebinding to the 𝗂𝗂𝗺 and putting a pause on it again. So that's a model of good inhibition. So yes, all inhibition is going to decrease the rate of cellular respiration, and a lot of the time that's not a good thing, and that can be very detrimental for the cell. But there are some specific examples where actually it's useful for the cell and helps make sure the cell is being efficient and not wasteful. Alright, so exam question 3 says rotenone is a chemical compound that's used as an insecticide and a pesticide, which is a substance that kills fish. The rotenone molecule disrupts the electron transport chain in animal cells by interfering with one of the essential reactions within the electron transport chain. Which one of the following statements best explains the effect of rotenone in causing the death in insects and fish? A. The rate of glycolysis would increase. B. ATP would accumulate in the mitochondria. C, aerobic respiration in the mitochondria would be disrupted, or D, the cell membrane would no longer be permeable to oxygen. So you can pause the video now as you go and answer it, and then check your answer in the video linked in the description box below. Now, with inhibitors that get introduced to a cell, it isn't the end of the world. And we've mentioned this before when we talked about cellular respiration, that if your inhibitor is competitive, then you can somewhat overcome its effect simply by increasing the amount of substrate present. If you keep increasing the substrate concentration, then that can help to sort of outcompete that inhibitor and allow that substrate to rebind to the active site. And that means that you'll overcome the effect of the inhibitor and the organism can return to its original rate of cellular respiration. However, if the inhibitor is non-competitive, then increasing substrate concentration won't do anything because the substrate is trying to attach to the active site, but there's nothing there. The non-competitive inhibitor is actually attached elsewhere, and so you can increase substrate concentration, but the maximum possible rate of reaction will be reduced thanks to that non-competitive inhibitor, and no amount of changing the substrate will have an impact on that. You'll actually have to get the non-competitive inhibitor detached some other way and removed from the system in some other fashion. So that is how you can actually overcome inhibitors. Now, this actually brings us to the end of this PowerPoint. If there was anything you didn't understand, you can go back and rewatch that section or reach out to your teacher for further clarification. Otherwise, I will see you in the next video. Bye.